Our speaker is Pires. He's an associate professor in biological sciences, and he is a researcher at the Life Sciences Building. He has a really cool story to tell us today, and he was interviewed by NPR just a few weeks ago, and he has some cool props over here for, very, very dinner themed, that hopefully, if you're craving some broccoli or cauliflower, artichokes, feel free to come and grab some after. All right.
Okay. Thank you. Can you hear me okay in the back? Yes. Okay. If I back away from the mic, just wave your hands. So today I'd like to talk about flavors. Like, why does the beer that you're smelling or the food that you're tasting have the flavors they have? And when you ask people why they think plants have flavors, you get really interesting answers. So I thought I would talk about that today, particularly how mustard and wasabi and horseradish have the really sharp, bitter taste, or the hops in the beer have bitter taste, or caffeine, coffee, and tea have bitter tastes. So the answer, of course, has to do with evolution. And I'm going to talk about two kinds of evolution today, coevolution and convergent evolution. So I'm going to define those later on. But let's just start, this is the audience participation part. Let's just start with thinking about evolution as you may have learned it in your high school biology class. So when you think of evolution, is there a particular person that comes to mind? Darwin. Okay, good, Darwin. What did this Darwin guy do? Finches. I love your one-word answers. So there's something about these finches, right? He got on this boat, the Beagle, he went around South America, he got to the Galapagos, and my friends who go there say, well, if you go to the Galapagos, evolution is so obvious, right? Because on the mainland in Ecuador, there are these wild-type birds that have a particular kind of beak, and then you go into the Galapagos, and the finches have all these different kinds of beaks that have different shapes, right? So the idea that there's selection for this variation seems kind of obvious. But how did he start his book? He started his book on experimental or natural selection via experimental evolution. So we're going to talk about vegetables in a second. But before I talk about human domestication, I want to talk about evolution over a longer time frame, like hundreds of millions of years. So when you taste bitter, what do you think that's for? It turns out that flies can taste bitter, jellyfish can taste bitter, all these things can taste bitter. Bitter is basically a self-defense mechanism, right? It's a signal that do not eat the bitter thing. And you put something really bitter in front of most people, it's a universal, what do they have? The universal yuck face. It's like one of those cross-cultural, I do not like that bitter thing. And yet we eat things like broccoli, mustard, and whatever, right? So there's a balance between bitter being disgusting and bitter being something pleasurable. So we talked about Darwin, and we talked about finches. So what are the two words? I already said them. What are the two words you think of when you think of Darwin? Natural selection, right? Survival of the fittest. But if you talk to somebody who really doesn't like the idea of evolution, are they upset by these two words, natural selection? Maybe, but really what they're upset about are two other words, common ancestry, right? Darwin's really radical idea is not that there's selection. The radical idea is that we're related to each other, right? That humans are related to primates and other animals, and these vegetables are related to other flowering plants and green algae, and that we're all joined by relationship to a common ancestor. So the story I want to tell, first of all, on the origin of mustards is a 90 to 100 million year old story. Now you can imagine shaking the hands of your parents and your grandparents, maybe your great-grandparents, but then after that you have to look up the family tree, right, and go all the way back. But you can imagine a handshake going all the way back to our primate ancestors and so forth. And so what my lab does for research, one of our research goals, is we make these family trees. Instead of just being the family trees among how we're related to each other, within a species, we look at how species are related to one another. So say how humans are related to chimps, are related to orangutans, or gorillas, to bats, to other mammals, all vertebrates, and so forth. And similarly, how these broccoli, cauliflower, Brussels sprouts are related to cotton or soybean, and so forth. And so in a study, we wanted to know how various members of this order, the Brassicales, which includes all these vegetables and other things all the way out to papaya, were related to one another. And how do you decide how things are related to each other now? Well, we used to figure out how things are related to each other by looking at morphology, right, and the anatomy of plants. Well, now we use DNA sequence, right? We can sequence genomes like that. Undergraduate research projects are sequencing genomes, right? So what we did was we looked at the evolutionary tree, what we call a phylogeny, of how the mustards and their ancient relatives are related. And what we did was we lined up that evolutionary tree with the bitter tastes that you enjoy in your mustard or in your horseradish or in your wasabi. And it turned out that that bitter taste comes from chemicals called glucosinolates. What are glucosinolates? Glucosinolates are compounds that contain nitrogen, and they occur in what we call a mustard oil bomb in the plants. So in plants, there's two cells, and it's kind of like a really bad like James Bond movie where you see like the bomb has like two parts and then like things mix together and then they explode. So there's basically two compartments in the plants, and they have the two parts of the bomb, and when a caterpillar or something bites the plant, the compounds mix together and become toxic, right? And then that's the bomb. So what we found was, when we looked at how these glucosinolates, and there's a lot of variation for them, mapped onto the phylogeny or the family tree of the plants, that there was a really interesting pattern of the glucosinolates becoming more and more complicated. So early in evolutionary time, there was, I'll call them simple glucosinolates, and then with the origin of the mustard family, there's more complicated glucosinolates, a fancier bomb, and then within the mustard family, there's even more elaborate mustard oil bombs. So we were going to publish this about 3 years ago, and a friend of mine who works on the butterflies from Sweden was traveling through town, and this is a great story about how science works as collaboration. So Team Butterfly meets Team Plant. And so the other researcher, Chris Wheat, was studying the butterflies, and he had just made his own evolutionary tree of the butterflies, the cabbage butterflies, that can eat these plants. So just remember, right, like this kale, for example, very leafy. When the ancestor of this evolved glucosinolates, suddenly other things couldn't eat it. Beetles couldn't eat it, all these various insects couldn't eat it, caterpillars couldn't eat it. But some caterpillar, after the first mustard oil bomb was invented, had some mutation that allowed it to eat this plant. And so what we did was we looked at the phylogeny of the butterflies. We said, oh, look, in this part of the phylogeny, the plants invent the mustard oil bomb. And in this part of the butterfly phylogeny, they invent a countermeasure, a way to diffuse the bomb. So how does that work? They have a protein that not only allows them to eat the mustard oil, but also they can turn the mustard oil into its own defense mechanism. Sort of like a monarch butterfly is poisonous because it can eat a milkweed. Well, the same thing is true of cabbage butterflies. They have these white colors that aren't camouflagey, right? They're basically signaling, I am also poisonous. So what was really interesting was we found 3 such radiations or evolutionary give and takes where the plant evolved a mustard oil bomb and the insect evolved a countermeasure about 90 million years ago. And then about 50 million years ago, at the origin of the mustard family, the plants evolved a more elaborate mustard oil bomb, and the butterflies responded by evolving a more elaborate defense mechanism, and so both groups radiated. The plants radiated, and then also the butterflies radiated. And then that happened a 3rd time. So this process of this evolutionary fight is called coevolution. And Peter Raven and Paul Ehrlich, also a botanist and an entomologist, coined this term 50 years ago, long before genomics, because they knew that plants had these defenses and the butterflies had these countermeasures. But what they didn't know was what the evolutionary relationships were them and what the process was by which they got more complicated. So the other component of the study that we did was not only figuring out the timing of these events, but also figuring out what was the genetic underpinning of that. So you may think of traits as being like Mendel and single genes, and then alleles being, oh, I have blue eyes or brown eyes or green peas or yellow peas or round peas or wrinkled peas. But what we found was it wasn't alleles, it wasn't copies of a particular gene that gave rise to either the mustard oil bomb or the insect countermeasure. What it turned out was to be duplicates of genes, so extra copies of genes, right? And it turns out this is also very important for human diseases. So lots of human diseases, be they cancers or schizophrenia or Alzheimer's disease, are actually not caused by particular genes that are bad. They're caused by not having enough copies or having too many copies of different genes. But over evolutionary time, what can happen is that these extra copies of genes can be recruited to do other things, like making fancier mustard oil bombs or making the defense against it. Okay, so that's the first thing I wanted to discuss was how is it that you can have coevolution give you mustard, wasabi, horseradish, and so forth, and that you can thank your caterpillars for the condiments that are on your steak and so forth. Okay, the second thing I wanted to talk about was not over a 100 million year time span, but over a 10,000 year time span, how long that we have had to domesticate plants and animals. So back to Darwin. So it was great earlier when I said Darwin, you said Galapagos finches. But how does he open his book? If you read The Origin of Species, he doesn't start with the Galapagos. What does he start with? Pigeons. He starts with pigeons. So does anybody know about pigeon breeding? What do you breed for in pigeons? You breed for pigeons that have fancy crests. You breed for pigeons of all kinds of different colors. You breed pigeons, they have these things called muffs. Muffs are these giant feathers that come off the side of their legs, so they can't fly anymore. They basically have to do somersaults. So you breed for all these different pigeons. But the reason why he was talking about pigeons was because he knew his readers could understand evolution in real time, right? You could breed for pigeons. What's another good example? So when I get on an airplane, and people ask, what do you do? And I say, oh, I'm a professor at University of Missouri. And they say, what do you teach? And I say, I teach one class, but I do research on the dogs of the plant world. So dogs are, of course, phenomenal example of selection, right? But everybody knows that a Chihuahua and a Great Dane are a dog, right? Even though a Chihuahua and a Great Dane look really different. So what I tell people is this. Do they know that kale, cabbage, Brussels sprouts, kind of like little cabbages, but not really. Broccoli, cauliflower, and I didn't find one in the store, but also kohlrabi. This is one species. Okay, who knew that that was one species? 1, 2, okay. Not too many people, right? Why is that? Why is it so hard to imagine this is one species? I think in part because when we think of domestication, we usually think of things getting bigger, right? You think of, oh, if I go to South America and I find a wild tomato, the tomato is like this big. But if I show you the California Flavor Saver tomato that's this big, you still know it's a tomato, right? Or if I showed you ancestral corn, okay, it's got 8 rows and it's this big, but it still kind of looks like corn, unlike its wild ancestor teosinte. But you show people all these vegetables, and there's no way that they know it's one species, right? Because what have we done? So what's the part that we eat here? We eat the leaves. So what have we done? So the wild ancestor of these, which I also collect and grow and have sequenced, grow on the cliffs of the Mediterranean, and they have leaves, but they're not curly like this. They don't have the same chemicals in here that we've selected for. And they're biennial plants growing in the Mediterranean. So here we've selected for leaves. What have we selected for here? Also leaves, but something else. We've selected for it not to flower, at least not very fast, right? Like you have to put these in the winter to get them to flower, right? So this won't flower, like this is it. You'll harvest this thing. What are these? Brussels sprouts. What are they though? They're leaves, but they're from the buds of a plant. So actually, on a plant, if you have a stem and the leaf comes out like this, the bud is this part. So they look like little cabbages, but they're actually convergent with cabbages, right? These are actually not leaves like that. They're from the bud. What about this? These are the sexy bits, right? So these are the vegetative parts of the plants. So these are little tiny flowers, right? And then when they flower, when the real flower, like, do you think these flowers are the ones that open? No, when the plant reproduces, it sends up another stalk that comes out. And what about this thing? Does anybody know what part this is? So if you break them open, so there are flowers, but there's a structure called a curd inside that we eat. Curd, K-U-R-D. But it's part of the inflorescence. It's also part of the sexy bits of the plants. But again, when they want to reproduce, they don't reproduce from these, they send up another flower stalk. So who has never ever liked these? Broccoli? Everybody in here likes broccoli and Brussels sprouts? Okay, sample size is not big enough. So people used to think that taste was something that was totally environmental, right? If you didn't like your broccoli and Brussels sprouts, your mom would tell you to eat them regardless because they were good for you, and it was something wrong with you. But it turned out in the 30s and 40s, people started to find out that actually taste is the most variable sense that humans have, right? There's very strong selection for eyesight and for being able to touch and to hear, right? So if somebody's disabled, usually they're disabled if they don't have their eyesight or if they don't have their hearing. But if you can't smell or taste, is that really disabled? Yes. But it turns out that bitterness, being able to taste bitterness, is very genetically controlled. And so if somebody says they don't like broccoli, like George Bush, the senior, who said on Air Force One in 1990, I'm not going to eat any more broccoli. My mom made me eat broccoli, and I'm not going to eat broccoli anymore. And if you asked him if he liked Brussels sprouts or cauliflower, he would say no also, right? Because they're linked. So anyway, so these also have these tastes, and we've now selected for them. So this is also related to these glucosinolates that cause this bitterness are also related to canola oil. So does anybody know that canola, like the term laser is an acronym, right? Light amplification stimulated by the emission of radiation. Did you know canola is also an acronym? What's it stand for? Do you know? Canadian oil. Canola is Canadian oil. So what did the Canadians figure out to make Canadian oil? How to make oil that is not bitter, basically, right? That's low in glucosinolates, but high in the other parts that we like. Okay. So last story about bitterness is, because Jason wanted it, was caffeine. So what things have caffeine in them? Coffee. What else? Chocolate. Tea. Coffee, chocolate, tea. Do you think those three things are related to each other? So if they're related to each other, that'd be very easy, right? You can say that the origin of caffeine evolved once, right? But it turns out that coffee, chocolate, and tea are not related to each other. So what does that mean about caffeine? That has evolved 3 times. So in the last couple years, we have now sequenced the genomes of coffee, chocolate, and caffeine. And when the coffee genome came out last year, they actually wrote, the reason why the paper got such press was because it was on the convergent evolution of caffeine in those 3 crops. So the one kind of bitterness that we enjoy in these vegetables is due to coevolution with the caterpillars and their relatives. And the caffeine that we enjoy in coffee, tea, and chocolate is by convergent evolution, right? That it evolved more than once. Like the hops in your beer that are bitter are not because they make us happy, but because these are the plant's ways to defend themselves against microbes and insects and fungi, and that we have now secondarily learned to enjoy them. as condiments. Thank you. Questions?
So we have time for questions. We have 2 microphones, one in the back. I have the other one, so you can just raise your hand and we'll just come to you when you have some questions.
So if these things are toxic to insects, why aren't they toxic to us?
That's a good question. So Heidi wants to know if they're toxic to insects, why are they not toxic to us? So obviously mammals and birds have a different battery of ways to digest things than do the insects. I suppose I can eat these raw, but of course I prefer to cook them. So in the process of cooking and fermenting, the invention of cooking and fermentation is basically to break down things that we wouldn't otherwise eat or would have a difficulty eating. But that's a good question. I assume it's because they're tuned to the insect nervous system and not ours. Do you know? Is that right?
Well, what if your entire diet?
Yeah, I don't think it would be good to have a whole diet. So actually, I'm glad you brought that up. So bitterness, actually, so I said in the start that bitterness evolved as an alarm, right? Don't eat this rotting garbage, right? So how do we usually enjoy bitterness? Like take your chocolate. How many people actually eat raw cacao? You don't. So, you do if you're the Aztecs, or you mix it in your mole and make something that is spicy and bitter. But usually you take bitter and mix it with other things, right? So with chocolate, we mix bitter with sugary and sweet and milky, right? And with caffeine, well, some of us drink our caffeine straight, but a lot of people, I think, have caffeine with their milk and sugar, as far as I can tell, right? Or their tea. So usually you mix bitter with other things. And I like to eat these, but I don't think my wife likes me to eat a whole bunch. So yeah, you can't eat too many of these. What else? Cheryl's got a question.
So you were talking about duplicate copies or whatever. What might be is that relatable to humans, mammals that do the same thing?
Okay, good question. So how do you get duplicate genes? So there's a couple ways. One way, which is what happened in the case of the cabbage butterflies, is you have a gene and basically there's a cut and paste error and you make a gene that's right next to it. Or do you remember why Barbara McClintock got the Nobel Prize for her work? Because she discovered jumping genes, right? So basically pieces of selfish DNA move pieces of genes across the genome to someplace else. It's kind of a natural GMO, if you will. So there are these kind of genes where you're just duplicating one gene or a couple of genes at a time. In terms of the plants, though, they do something called polyploidy, or whole genome duplication. They also do the tandem duplicates, but in the case of mustard oils, they evolve by polyploidy. So what happens is the whole organism doubles their number of chromosomes, and then over evolutionary time, they lose most of their genes, but they're able to keep some genes that are perfectly retained because for selection or whatever. The advantage of polyploidy is imagine that you have a very complex network that's required to make something like an eyeball. Suddenly you have all the genes to make an eyeball duplicated, and now you can make something really cool, right? Because you have all the extra copies of these things that, eyeball is not a good example, but flower petals is a great example. Or the case of the mustard oils, the glucosinolates. So what happens is you duplicate the entire pathway that makes the mustard oil bomb, the glucosinolates, and then you can lose many copies of it, but you keep all the genes that are very important for signaling and transcription. Does that answer your question? No, I failed.
Well, I needed that, but is there a human equivalent?
Oh, humans, sorry, yes. So vertebrates don't go polyploid too often, although all vertebrates share two polyploid events. But because most mammals, for example, have sex chromosomes, it's hard to go polyploid, right, when you have X's and Y's, right, because of the duplicating sex chromosomes. But there are So polyploidy is fairly rare, but it does happen. Aneuploidy also happens, right? Where you get extra chromosomes. But extra chromosomes is usually not a good idea, right? You get Down syndrome or other things. But this idea of genes being duplicated happens all the time. So people talk about that as copy number variation. And so now this turns out to be very important because originally people would take the original human genome and they would just compare sequences to the reference human genome. But what would you miss? You would miss anything not in the reference genome, right? So if you had an extra copy of some gene or some new gene, you wouldn't detect it, right? And so now that's why there's all these 1,000 genomes project, sequencing all these humans, or 1,000 Arabidopsis plants or 1,000 maize plants, so we can find exactly all these duplicate genes and find out what they're doing. And like I said, a lot of human diseases like Alzheimer's and schizophrenia, whatever, are not caused by alleles, but by the incorrect number, different copy numbers of genes. Also things like our ability to digest starch, like to eat bread, right? Or for your dog to eat bread. What is that? That's extra copies of the genes that digest amylase, or the sugar digesting genes are extra copies of genes. Or why some of your friends may be more alcohol tolerant than others, right? Because they can consume alcohol or not. Any other questions I can't answer?
I have a question.
Oh, sure, Jason.
So you said that there were 3, I think you said radiations.
Yes.
Plants developing
Yeah.
mustard bombs and
Yeah.
insects overcoming mustard
Yeah.
bombs. And you said they were like 100 million years ago, 50 million years ago, whatever it was.
Yeah.
How do you know it was 100 million years
Okay,
ago? How do you have an idea
good
of
question.
how long it might be?
Okay, very good question. So you have to have 3 things. You have to have a phylogeny. You have to have some kind of clock, which we can estimate from molecular DNA sequences. And then you have to have fossils, right? So it turns out, do you think these make good fossils? Not so much, right? Because they're leafy. But there's relatives of these that are woody, that leave fossils, right? So in our analysis, we included things that were woody. And then same thing on the insect side, right? We had some butterflies and some other things that also had fossils. So if you have some fossils, you can then constrain your phylogeny to say, okay, these 2 things branched at this time. Of course, you would love to have a piece of amber from Jurassic Park with a caterpillar munching on something like this, but we don't have that. But we do have good fossils among related plants to these, as well as within the insects to time the tree. And those are also minimum ages, right? So if you have a fossil, you know that it's at least that old, but of course it could be older, right? At least a little bit older.
I have another question. When you study that, when you look at the fossils and all that stuff, how do you measure? Like, how do you measure how old?
Okay,
it
good.
is?
So we spent a year on the first figure of this paper. So I told you that we lined up the evolutionary tree of the plants with the evolutionary tree of the butterflies, right? To say that when this split, these butterflies radiated. So each of those measurements has an error bar on it, right? And so our story doesn't depend on it's 90 years plus or minus 5 years, right? It's 90 years plus or minus 10 million years, right? Or whatever, right? And also, we didn't really focus so much on the exact timing of the events. It was more that we could tell that this event happened first in the plants, and then this happened in the insects sometime later, and then this happened in the plants, and then this happened. So that's what we were able to unravel. If somebody tomorrow comes and says, oh, it was 120 million years and not 90, I'd be like, awesome, glad to know.
When you look at how many
Glucosinolates.
Are on what you were studying,
Uh-huh.
how do you look for them when you look at?
Okay, so again, this is team science, right? And so we're not going back in time and looking at what the ancestors had. What we're doing is we're looking at the related wild species and even more distantly related families. So things like papaya and a bunch of little families that you've never heard of, like meadowfoam, and there's an African tree called moringa. And other people had measured the current existing species for their metabolic profiles for glucosinolates, right? And so what we can say is, well, based on what these lineages have today, we can estimate that this branch of the tree had these type of glucosinolates, and then when you get to these guys at this part of the tree, they have these more elaborate glucosinolates, right? So we can estimate the order by which they evolved, right?
Look at the tips of branches.
Yes, so we look at the tips, we look at present day species to infer each of the ancestral nodes for what their glucosinolate ability would be. There's a microphone coming.
You've mentioned several times collaborative science. When you were in grad school, you probably didn't think you would collaborate with some of the disciplines that you are now.
Yeah.
And so, what disciplines do you collaborate with? You mentioned entomologists. And your life sciences botanist, I guess?
Yeah, so
Probably a statistician.
yeah, I love the gray space of hybrids. I study hybrids. I'm a hybrid, right? We're all hybrid, we're all eukaryotes. So I think now, like when scientists are trained, right, like undergraduates have to learn computer science and math and statistics, and right? I mean, the undergraduates in the lab are often double majoring in computer science or math. And then they go to grad school and they learn something in detail, and then they go do a postdoc and learn some other skills, right? And so, you go from being, when I was a graduate student, I said I was a botanist. And then when I was a postdoc, I said I was a genome biologist. And now I do collaborate with people who are in statistics. Or we have another paper I didn't talk about today, but to discover the origins of these, we were collaborating with people on linguistics, right? What are the names of these things? And I'm not going to, yeah. So anyway, you're not only collaborating with people in other sciences, but also people in the social sciences and anthropology and so forth, if you're talking about some of these things.
How do you make those connections? Science cafes?
Yeah, I guess so. Maybe somebody might come up afterwards. I mean, I go to meetings. I read a lot of articles. I find a lot of stuff on Twitter. Whatever it is. So, and my students go out and make connections. Right? I encourage them to go out and find. If the lab is limited by my brain, then that's too bad. I like I like other people to expand it.
So if you were going to help farmers that were into growing your type of veggies, what direction might that look like? I mean, what could you do for them?
Well, I've failed to get any grant that has any practical value, so I don't think I'm the right person to ask. But like I said before about canola oil and other things, people do select for levels of glucosinolates, for example, right? I'm actually quite interested right now in how would you make, so for example, kale. There's a kale that's grown in Africa that a lot of people eat on Fridays because it's the end of the week. As they run out of their meats, they eat their kales to kind of get by. It's like a stew they make at the end of the week. So I've been thinking about, with some collaborators in Cornell and other places, how could you make this have more vitamin A, vitamin B, vitamin C, right? And how could you make a heart-healthy kale that would be good to eat at the end of the week? And then maybe Gates Foundation will pay for it or something like that. I don't know. But I usually don't wake up in the morning thinking about how to feed the world, although I think the research we do would be able to help somebody who's a plant breeder who wanted to do that. The problem living in Missouri is a lot of people don't grow these here, right? I mean, the big markets for these crops are in California, Oregon, Washington, Michigan, New York. Now, if you go to the South, they do have, what do you make with your pork? Collards, right? So, like, collards are a crop that, and one can't, I mean, you can see things like this in the farmer's market. The other problem is that a lot of people get their seeds from Europe. And so there's not like, they've been working with European seeds. So we've been trying to figure out how we can make, I mean, if you go to the farmer's market, you basically see this kind of kale and what's called dinosaur kale. That's like two kinds of kale. I have like seeds for hundreds, if not thousands of different kinds of kale. So, Maybe I need a sexy purple and orange kale leaf or whatever, or something like that, that I can sell at some elite farmer's market. I don't know. I obviously have not thought too much about the practical side of it, but that's okay.
Well, I guess going back to a little bit of the science, you mentioned that flies and other animals are also able to taste bitterness.
Yes, but
How do you know that?
that's because I read it. It's not my area of research. When I was looking at bitterness before, like 2 hours ago, I ran across that, so I know that that's true, but I would have to give you the reference. I don't know the original source off the top of my head.
Any more questions?
No?
All
All right, thank you very much.
All right, well, thank you. I'd just like to remind you that we have a Science Cafe for next month. It will be on September 4th, the second Monday of the month, and